Thank you. And in the tradition of NASA-Ames Research Center, before welcoming you, I'll say, now that we all understand the safety, we could go on with this talk today. I think one of the things that I do want to point out, you are here at NASA-Ames Research Center, and the connection to biology is really critical, because one of the things that we do here is, besides all the space exploration and all those different fascinating robotics missions that you see, what Ames Research does is also concentrate on the life sciences areas. And so why space? Well, you could think about it in multiple areas. It's where we began, so it tells us our history. It also is where we are right now, and where it teaches us understanding about life and how life evolved with a gravitational field. And it is also the future. Some of the talks, both talks that you're going to hear today, will cover the origins of life. That is really important. How did life begin? Is it anywhere else in the universe? We know for some of the missions that we've done here at NASA-Ames that there's water everywhere, that there are habitable planets out there with the Kepler mission. We're getting to see insight on the building blocks that exist for life everywhere around us. So the question is, what was the beginning? Was it a DNA world? Was it a protein world? Was it an RNA world? How common is life? What is needed for that origins of life? So Ames concentrates in that area, pushes the envelope in that area, changes the way we see that. And besides, it's an exciting place to be at, and that's why I'm here. I'm not here to give a talk. I'm here to just welcome you to the center. So with that, I will end, and I will introduce Tucker Hyatt from the Wonderfest, who, along with the Commonwealth, are putting this event together. So again, welcome to NASA-Ames.
Good evening, and welcome to our program. The program is hosted by the Commonwealth Club of Silicon Valley and by Wonderfest, the Bay Area Beacon of Science. My name is Tucker Hyatt. I'm the founding director of Wonderfest. Right off the bat, I'd like to thank and welcome our Commonwealth Club members and our Wonderfest supporters. For those of you who are not members or supporters, I encourage you to learn more at the materials table back there and at our respective websites, commonwealthclub.org and wonderfest.org. Your involvement will support nearly 100 events held each year in Silicon Valley and the greater Bay Area. Tonight's program has been a long time in coming. Its planning began last June, but certain government venue funding issues caused delays. Now, we're very glad to be here and hope you're ready for a terrific presentation. Toward the end of our program, we will have time for your questions. Please just raise your hand and our wonderful volunteers will bring the microphone to you. Now, we ask you please to all silence your cell phones, and please join me very soon with some applause in welcoming our two speakers. It is my pleasure to introduce Dr. Lynn Rothschild, astrobiologist with the NASA-Ames Research Center, and Dr. Richard Zare, professor of chemistry at Stanford. Lynn Rothschild studies biology, in particular astrobiology and synthetic biology, here at NASA-Ames. She has degrees from Yale, from Indiana University, and a PhD from Brown in molecular and cell biology. Her fieldwork has reached from Australia to Africa to the Andes, and from the ocean floor to 100,000 feet in a balloon. Dr. Rothschild is a fellow of the Linnean Society of London, the Explorers Club, and the California Academy of Sciences. Richard Zare is professor in natural science. Besides chemistry, his greater field is professor in natural science at Stanford University. He is a graduate of Harvard, where he received his BA degree in chemistry and physics and his PhD in chemical physics in 1964. Dr. Zare has held professorships at MIT, at Columbia, at the University of Colorado, and since 1977 at Stanford. Professor Zare's many, many awards include the 2008 Pimentel Award in Chemical Education and the 2010 Priestley Medal, the highest award given by the American Chemical Society. Ladies and gentlemen, please join me in welcoming Dr. Lynn Rothschild and our first speaker, Professor Richard Zare.
Thank you, Tucker. Can everyone hear me? Yeah. Great. Okay, good. We're going to talk about I'm going to at least try to tell you about what can chemists tell us about the origin of life. And the answer, of course, is going to be not much, right? We know that already. And let's begin. Let's see, there's a clicker that controls the slides. Here they are. Okay. So it seems that chemists, lots of hubris here. They claim that they can make anything. They're able to make any molecule, okay? If chemists are so smart, then why don't they make life, right? And indeed, I'm hoping, perhaps in our lifetimes, we'll see something like that happen, which may have nothing to do with how life itself started, but will be life. We'll see. We have to, of course, try to define all this. So let's begin. The big question, as you heard from Chief Scientist Jacobs Cohn, was, are we alone? Always been a question that people have had and wondered about. And we next led to ask this question, how did life start on Earth? Because that's where we are. And to even approach this question, we got to ask something even more fundamental, which is, what is life? Okay? This is not, actually, this is not an easy topic at all. It's very hard to define if life. Now, some people think that there's life and non-life, and others think there's, a gray area in between. And people are still arguing about certain characteristics of things. For example, is a virus alive or dead? Okay, things of this sort. We can discuss this. Well, how are we going to define all this? What do we mean by life? And I think of it as respond to stimuli, okay, and a capacity to grow, and the capacity to reproduce. Believe it, these three, I assure you, this is not enough to define what life is. Because if you just accepted these three, you would decide that the stars are alive. They have a whole life cycle of the stars. And people don't say, no, that's not alive. So we need something else. And it's driven by chemical energy, not nuclear energy. Otherwise, we're stuck with all types of other things. Okay? So that's our picture. In fact, what we know about life, basically, at the moment, is what we see on Earth. So if it doesn't look like something on Earth, we don't know quite sure what to say about it. Yes, it should fit these. But we can even find examples of things which don't fit this, which we claim is alive. For example, if I remember right, trying to remember, is it a cross between a mule and a horse that gives you a donkey? Okay, or is it a donkey and a horse? Mule. Okay, so thank you. Horse and a donkey gives you a mule, and we think of mules being alive, but mules can't reproduce, right? So we got problems already with our definitions, just so we understand how tricky this business is. So how did life begin on Earth? Well, there's been all types of thoughts about this for a long time. And the first is various supernatural explanations, various creation myths. All types of religions have their own creation myths. And they're fun and they're interesting. And if this is what you believe, why are you in this room? Because you know the answer already. Okay? And just, so that part is the way that is. And that's not where it is for me. I'm still puzzled. So next thing is some form of spontaneous generation. Somehow you need to go from not being alive to alive. Okay? So that's, so it has to be some type of natural process. Now there's a third idea that's very popular. It starts with a Swede, a famous Swedish chemist by the name of Arrhenius. And it is called panspermia. And it is the idea that it all started somewhere else and came to Earth, which may be true. But let me tell you, this is an uninteresting explanation. Because, why? Because it doesn't really answer the question, where did it start somewhere else? So we still have to think about how you go from non-life to life. Or you believe that things were always alive. Okay? And that's another problem. Big Bang and all, okay? So that doesn't make sense either from what we know. So somewhere we got to face this and come up with some explanation for this. Well, people have thought about this. One of the brilliant people in this matter has been Darwin. And Darwin put forward the idea of warm little pond. Okay? And I think I have a quote from him. I hope it's on the next thing here. And I'm going to read it to you as best I can. Okay? It's often said that all the conditions for the first production of a living organism are now present, which could have ever been present. But if, oh, what a big if, we could conceive in some warm little pond with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed, ready to undergo still more complex changes, at the present day such matter would be instantly devoured or absorbed, which would not have been the case before living creatures were formed. And this was Darwin's letter to someone in 1871. Interesting. Okay. So we have that type of picture. That's been around. Then we go to a next picture. Whether or not there's something called the primordial broth. You know, Campbell should be so lucky, right? And here we go. And in 1953, Miller and Urey synthesized amino acids by passing sparks or lightning through a gas mixture of ammonia, methane above a pool of water. Okay, here's very much the chamber of what they did. Classic experiment, really. Very exciting. When I was growing up, my mother told me that's how life started. Okay? And here's a boiling flask in Earth's primitive oceans, a cloud formation, primitive atmosphere, and sparks, power supply. Okay? Some picture like this. And I don't believe this at all anymore. Okay? I don't, and I'm not trying to give you arguments why this is not. It may be part of what goes on, but no, it's not the answer to me. And I'll try to explain why I'm not enthusiastic about this. So there we are. There's Stanley Miller's prebiotic soup. Just for example, it turns out that if you have carbon dioxide around, which you expect to have in early Earth, you don't get these amino acids, and you got all types of problems as to what's going on. Okay, so what can the historical record teach us? That'll be the next question. And things we call life are so amazingly complex, yet for centuries people believed that it simply came out of nothing. This was historically so. A wonder unbidden. From mud, serpents slithered away. From rotten meat, maggots sprang forth, as shown here. Okay? Bad. Well, it's after dinner. It's okay. And that was thought to be the case. And then a brilliant scientist put an end to this. This was called spontaneous generation. And who killed it? Pasteur. By the process we now call what? Pasteurization or being sterilization, right? And life, and what Pasteur did is he scrupulously kept things clean. And he found that you could take broth and nothing would come out of the broth. It was it. That was it. Nothing happened. And then Pasteur claimed, who was a very good Roman Catholic, that life only came from life. And that was accepted. well, as you see, that idea of life only coming from life and fine, it leads us back to this problem. I don't believe that. You have to go somewhere from non-life to life. But as I say, the geological record argues forcefully otherwise. Pasteur is not right in saying that, but we still don't know the answer. So now setting the stage. What can we find out? Let's begin. The story of life begins with the origin of the solar system, right? And here we are, you know, the formation of a star condensing out of a cloud, and we end up with all types of things like a molten Earth. Well, molten Earth is not a place that we recognize there's going to be any life. It's going to be, you know, destroyed. You can't heat things up to 500 degrees centigrade or whatever and expect to get any life. That's it. So some like it hot. Here we go. Various things we know about the planets and where they're like. Whatever are the conditions of life, it seems at this point, at least to me, that they're rare, not, how do I say, ubiquitous. Now, some people think it should be everywhere, and there's a story, as you've heard, you should just look for water and look for carbon, and then you'll get habitable planets and you'll find life. Doesn't seem to me to be likely at all to be the truth. The SETI experiment, trying to find out whether there's intelligent life, that's another matter somewhere. It's not obvious at all that there's anything you can find in quite a distance away. We just don't know, okay? But here's what we do know about life. All we have are the fossil record and the oldest rocks dated on Earth. And we can date things back to about 4.6 billion years ago. We can go through the whole story of radioactivity and various forms of dating. And, you know, plus or minus 0.1 billion years. It's really good. Okay, don't have to do better than that. But anyways, as you go on through this, you'll find that the earliest form of life is single cell. And you expect that. In fact, you might not realize it, we're made up ourselves of huge amounts of single cell. That's how we digest. Okay, we're loaded with single cells in us. It's very necessary. And single cells remain on Earth the still most common form of life. Okay? People are always busy talking to you about diversity, you know, and they're worried about various furry creatures disappearing. Well, turns out the real diversity is bacteria, and they don't talk about that. Okay, we'll continue. We'll go back. Let's go back. Let's look through this through. 2 billion years, evolution of cells with a nucleus. It takes quite a while. Life seems to start as soon as we can, quite early on. It takes a while to evolve and get to multicells. We don't even understand how single cells fuse to form multicells for sure, but then it takes off into higher forms, let alone bones. Bones are great for fossils. Cells are miserable, right? Because they degrade, they fall apart. They have a lot of trouble in this. Here's other ideas. This is due to David Deamer, a friend of mine, a brilliant man who's at University of California at Santa Cruz. And this is called the bioenergetic perspective. And he thinks of these various infalls of material and forming vesicles, forming something that has an inside and an outside because you need a cell. And this goes on with condensing agents. And it's a picture he has of how you get some type of first cellular life. But we don't know much more about this. And his picture is that it comes primarily from some type of something coming to Earth, asteroid, comet that is left over. And it may be so, okay? We don't know. And then people talk about the RNA world. And I'm just reminding you what that's made of. Because we need, as we understand life, we need either RNA or DNA or both, right? We need some genetic code for it to continue. And you'll see that there are things that are sugars, and there are phosphate groups with this P over here that connect these with a backbone. And then there's these various bases that are made up of carbon and nitrogen and oxygen, like so, as shown here. And we need that type of stuff, of one sort or another, okay? And so in 1980s, Thomas Cech And Sidney Altman showed that some RNA molecules can act as enzyme-like catalysts and can promote making more of things. That was very exciting when you got to that possibility. I'll remind you that a lot of chemistry just doesn't take place at any reasonable time scale unless you get a catalyst to make things go faster. And the fact that you can get both DNA and RNA and act as a catalyst, which is normally a protein, very exciting possibilities. And that's led to lots of comments. And here's a division of opinion that I wanted to call to your attention. What is it? I read it to you. Scientists interested in the origins of life seem to divide neatly into two classes. The first, usually but not always molecular biologists, believe that RNA must have been the first replicating molecule, and that chemists are exaggerating the difficulties of what they call base or nucleotide synthesis, of putting carbon and nitrogen together. Okay? The second group of scientists are much more pessimistic. They believe that the de novo appearance of these oligonucleotides on the primitive Earth would have been a near miracle. The authors subscribe to this latter view, and they're Joyce and Orgel, who's at Scripps in La Jolla, okay? Writing on the second edition of the RNA World, 1999. And time will tell which is correct, or maybe neither's correct, as we'll see in a moment. Okay, there's more to think about this. And okay, so how did life begin on Earth? And here we go. We're looking for something in here to start this off before we get to all types of other things. Human life is really a short part of this, you know, the record, just so we all get a sense of that. Okay? The details of the beginning of life on Earth remain truly obscure. Scientists also believe that all life has a common origin, because when they've looked at all life, we see we can make a tree of life very clearly. What things seem to have come from other things and topics of evolution and so forth. All seem to have common things that are preserved that make up life. So whatever it is, once it got going, it really got going, but, and it seems to be all related. Fascinating, okay? So how to search for origins? That's one of the questions we're gonna face. And let's consider the automotive transportation system in the United States. And let's ask ourselves, what's the origin of that transportation system? Okay? If you wanted to explain the system in all its complexity, including, you know, 101 and how hard it is to get here, you would not ask whether cars led to roads or roads led to cars. That would not be the right way to look at this problem. It would be more productive to consider the state of transport in pre-industrial United States and ask how primitive foot and horse trails that must certainly have existed had developed into paved roads and so on. So it's interesting. We have to go back and think about it maybe another way. So maybe we don't want to get hung up on whether it's proteins first or RNA first. They have to somehow be together and work for things to go on and do something. Okay? And the picture I'm just showing you of this. And now we have all these interstates. So to understand the origin of life, we need to look for multi-step processes with feedback which may have occurred in the past. We want something of this sort that can amplify and build on itself. What to do? And I thought a very fascinating idea was put forward by this Australian, I think it's J.V. Smith and co-workers. They've suggested the uptake of organic molecules within the micron-sized, micron really small, right? Three-dimensional cross-linked network of pores found to exist within the top 50 microns or so of weathered feldspar surfaces. So you look at these rocks, and they're very porous, okay? And a 2.5 square kilometer granite reef is estimated to contain, you ready? 10 to the 18th, okay? 10 to the 9th is a billion, 10 times itself. A billion times a billion is 10 to the 18th. Catalytic microreactors, opened by diffusion to the dynamic reservoir of organic molecules, but protected from the dispersive effects of flow and convection, as well as protected from the high flux of ultraviolet radiation impinging on the early Earth. If you're on the top of the early Earth, you're in trouble. And I'm going to try to explain why you're in trouble. You're going to get hit on the head. You're going to get hit in the head by UV, which is going to tear you apart. And you're going to get hit in the head because all types of meteors and comets and asteroids are coming to the Earth. Not so good to be there. You really want to hide somewhere if you can. Okay? And I'm showing you the nature of this feldspar. And these are scanning electron microscope images of weathered surfaces of alkali feldspar fragments. And the scale bar that you see here is 5 microns. And fragment A from gravel, showing the beginning of formation of etch pits. Okay? B, a highly weathered feldspar from a peat soil inhabited by at least 3 varieties of rod and disc-shaped bacteria, which you can make out here. Okay? This is Earth now. It's so interesting, as you'll see, and as you've heard a little bit, NASA keeps pointing to space. And I was asked a while ago where we should look for life, and I suggested between your toes. And what I meant by that was not literally between your toes, but look down, not up. Okay? And this is, unfortunately, I'm not sure NASA has thought this is part of their mission yet, but we'll see. Now I'm talking about not platonic love, but plutonic love. Okay? The love of rocks and such. And in 1981, Frank Westheimer stresses the important and universal role of phosphorus in biochemistry. And you've heard so much already about carbon, and carbon's versatile for forming networks, much more versatile than silicon, for example. And we know about water. We've all heard about that. But not enough attention is given to something else. You need various other atoms. We know about nitrogen, but people pay not so much attention to phosphorus. Now, where's phosphorus? Well, it's certainly in RNA and DNA. You've seen that. And where else is it? It's in something called ATP, okay, adenine triphosphate. And without ATP, there's no motion. There's no way to do the chemistry that drives life. So it's in your mitochondria to run things, so you really need phosphorus. Okay? And this fact hints, I think, at the origin of life as life on the rocks. Okay? And I've pointed now to phosphorus for you. I want to also tell you about another element. Do not forget about sulfur. Okay? Well, the devil, it's sulfur. Okay, but what do I mean by sulfur? Why am I pushing sulfur to you? 2 of the essential amino acids, cysteine and methionine, contain sulfur. And now think about it for a moment. Methionine turns out to be one of the amino acids which is in every protein. Okay, part of our start-stop codes and all this. Methionine's there, and that means if it's in every protein, there's not only sulfur in every protein, but all the enzymes must contain sulfur too. Now, that's important. Phosphorus and sulfur, keep this in mind. Now I'm trying to lead to something for you. May I try? Do you know about any phosphorus in the atmosphere? No, I don't either. Okay? It just isn't around. Okay? You don't find it in the atmosphere. There just aren't good gases of phosphorus. There is something called phosphine, but you know, it's not going to work. Forget it. It's not going to work. Okay. And what about sulfur? Well, we know rotten eggs, H2S, but by and large, there's no sulfur. SO2 from coal-fired plant, you know, various things, but by and large, not much sulfur at all. Well, let's stop a moment and push ourselves another way. What about going to our warm little pond? Do you think that there's much phosphorus or sulfur in them? Turns out, I'm telling you chemist viewpoint, most phosphates are insoluble. And sulfur in the form of sulfides are insoluble. You don't have any concentration to speak of. And you don't want to go bigger than a little pond, because if you go in the ocean, you've diluted everything. Nothing will ever come together in an easy way. So you're stuck. Where are you going to find this material? Okay? You need heat. And heat drives me to what? To geothermal energy to a volcano. I need some form of heat. And where do we have this? One of the places is hydrothermal deep sea vents. And I'm going back to my statement again about looking down. This is very interesting, because you'll find in the ocean where there's tectonic plate action, and you really get very hot things. This is why they're called black smokers that come out and precipitate here. And you get all types of ecoforms in ecology going on, which we don't understand much about. It's hard to get to these places, and people haven't studied it much. A hydrothermal origin of life within porous, weathered granitic materials potentially solves an apparent contradiction. Polyphosphates in biochemistry are ubiquitous, as contrasted both to the relative rarity of crustal phosphate and to the insolubility of native calcium phosphate, okay, and consequent low average concentration of dissolved oceanic phosphate. But put them in rocks, we got real possibilities here if we get volcanic action. We get HCN, we get which can make our carbon nitrogen. We got H2S, we got phosphorus, which can go on and do things. And so most phosphate in the primordial crust must have been sequestered and nearly insoluble. Calcium phosphates are basalts, and weathering produced only dissolved monomeric phosphate, but volatile P4O10 is known to be a component of volcanic gases. So does life require a volcanic eruption? I'm suggesting it may. It may really be the nature. So we have to look for things which can do that. And I go on to tell you how this thing undergoes a process called hydrolysis to form polyphosphates and so forth. And they can then polymerize. And this is well within the temperatures that you find within hydrothermal vent systems. So interesting possibilities. Now, I haven't given you a recipe at all, but I've given you some, to me, reasoning that suggests that we should be looking at hot rocks, okay, in contact with water. Okay. Part 2. Okay. And so life starts in weathered granite reefs near hydrothermal vents using organics from Earth and from space. Okay, and we can bring them in from space. It's a combination of things. This is a possible synthesis. This hypothesis solves problems of early Earth atmosphere composed of CO2 and N2, for which lightning won't produce any amino acids. Okay? Hostile surface bombardment and UV irradiation, capture and concentration of dilute organics and capture by vesicles. Okay? Locale for catalytic synthesis and polymerization. I need metals in chemistry to make catalysis. Metals are really necessary to make things happen. I got them in rocks. I don't got them in the atmosphere. I don't have them in the ocean the same way. They're too dilute. Okay? Scarcity of phosphorus in usable form. Source of energy to drive biogenic chemistry. The need for multitude of microreactors to promote rich evolutionary biogenesis. So here's some conclusions. I'm sorry I'm running on a bit. We may never know how life started on Earth. That's a fact. Okay? I accept that. And, you know, I think people who tell you that they know the answer are fooling themselves. Sorry. Okay? Maybe we have been asking the wrong question. The right question is, how can you go from non-life to life? What's the recipe? Okay? And this topic, once only a matter of total speculation, is entering a new era when simple hypotheses can be experimentally tested. We really can carry out experiments. And whether you call it astrobiology or whatever name you give to it, cosmochemistry, it's all the same, right? And it's a very exciting time to start to think about this. Well, sorry, you thought I was going to give you an answer to this? Here you are. And with that, I'm going to turn it over to Lynn. Thanks.
I usually start with a disclaimer that my worst grades probably ever in my entire academic career were in chemistry, and that's why it's particularly embarrassing to go right after Professor Zare, who I'm an enormous fan of. But that being said, I guess part of it is that I never really understood why you took chemistry in school. It was more sort of a way to weed out the pre-meds as far as I was concerned, but clearly it was because I didn't have Professor Zare as an instructor. And it was one of those things that, being an evolutionary biologist, that seemed much more interesting because things change over time. And then when I got to NASA some decades ago, you start to understand that life is much broader than just even the evolution of life on Earth, which is as rich and wonderful as it is. And chemistry was this sort of immortal static thing that, again, you sort of suffered through. And then, whoops.
I almost got it. I saw you sliding.
Thank you. Well done. Let's put Darwin back where he was. But then you realize that chemistry is not immortal either, that also has had an evolution, that you have no such thing as chemistry before the Big Bang. In fact, even after the Big Bang, the first elements you have are hydrogen, helium, and a little bit of lithium, and basically everything else has to go through a generation of stars and so on. And ultimately in chemistry you get to something far more interesting than any other chemistry, and that's of course life. But how do you get from this Big Bang to life, both on Earth or really anywhere else, is a lot of what astrobiology is all about. So that's sort of the broad picture, and if anyone wants to talk about those sorts of things during the questions, I would just love it. But what I'm going to try to do is confine myself to this question on the origin of life and give a little bit different perspective than Dick gave, but not enormously different. So here we are conferring, and re-emphasizing what Dick said. I was generous enough to let him go first, that really the origin of life was not an issue. It wasn't an issue for the church. It wasn't an issue for most people until fairly recently. And I'll start with Aristotle, who points out that some plants are generated from the seed of plants, whilst other plants are self-generated through the formation of some elemental principle similar to that of a seed, and animals do funky things. That's basically a summary of the rest. And then, you can then get into the cookbooks here, as Jean-Baptiste Helmont. To make mice, you may want to copy this down. You mix a piece of soiled cloth plus wheat, and you wait 21 days, and you'll get your mice. Now, I don't know what the exact ratios are. You may want to play with that at home. Once you get the whole mouse recipe down, you might want to start on scorpions. You apparently place basil. I don't know if it's sweet basil or not, but you can again play with that between 2 bricks, and you leave it in the sunlight. And then you too will have a house infested with scorpions. So it was really all very straightforward. It didn't contradict anything because that was human experience. And then Francesco Redi came along in 1668 and had the first one of these experiments that culminated in the ones that Dick mentioned of Pasteur, showing that in fact life needs to come from life. But of course that just begs the question, because at some point you need to get life from non-life. And so the rest of this, I'm going to go into a more scientific approach to this. So let's start with the question, really, what is evolution all about? And this really refers to one of my absolute favorite papers from all time. Is something that was published in Science in 1977 with the title Evolutionist Tinker. And the question is evolution an engineer? And here's an example of some microfluidics chambers. Or is it a tinkerer? And this is actually a wonderful piece of sculpture that I saw in Brazil a couple of weeks ago, and I've just been waiting to use it in a talk. So here's this whale that was put together by leftover gears and pieces of metal and so on. So is evolution an engineer or a tinkerer? Well, the answer is that it's a tinkerer. Evolution uses what's available. So if you sit back and you think, well, how come we have an appendix? That doesn't make any sense. It's because evolution is not an engineer. If you were starting from scratch, you probably would not design an appendix, not for humans. There are a lot of other things that we do that are pretty stupid that you really wouldn't do if you were doing this as an engineer. But again, evolution proceeds one step at a time as a tinker, so you have to start with what's there. A bird doesn't just decide, hey, I want to fly, let's grow some wings. You've got other bones in place that elongate. You've got feathers maybe there for thermal protection. There's a lot of evidence that tyrannosaurs were feathered. The idea of warm, fuzzy Tyrannosaurus rex is a little difficult to get your mind around, but that's what the latest evidence shows us. But then it turns out those feathers were very useful, not for tyrannosaurs, but for their descendants, the birds, to fly. So evolution uses what's available, like someone just playing around in their garage. So this means that you have to start with what you have. And the more I'm thinking about it, the more I realize it actually fits a scene in the movie Apollo 13 to a T. So if you'll indulge me, I'm just going to play this.
Okay, people, listen up. People upstairs handed us this one, and we got to come through. We got to find a way to make this fit into the hole for this, using nothing but that. Let's get it organized.
Okay, let's build a filter.
Better get some coffee going.
And that pretty much describes my vision of what you've got for the origin of life. You have to use what's there. So how is this really relevant? So in other words, to make life, and here's life. This is a wonderful video clip that one of my students did recently in our lab. And you can see the Bacillus subtilis dividing there and growing. This is, of course, a time-lapse image. They take about 30 minutes or so to divide each time. But you can only, to make this, use the periodic table. And as Dick pointed out, of course, you use a lot of carbon, you use a lot of oxygen, you use a lot of hydrogen, but you also need sulfur, phosphorus, and so on. So you've got those ingredients, and probably water is a solvent. There are all sorts of great reasons. It's widespread. It's got lots of great chemical properties. And one that's probably not quite so obvious, but it's important if you're going to look for life elsewhere, and that is that water's unusual in that when it's solid, it floats. And so think about the last time you had a gin and tonic. Normally that would have been last summer, but with the weather we've been having, it could have been yesterday for all I know. What happened to the ice cubes in the gin and tonic? Did they sink at the bottom of the cup, or did they float? Of course, they floated. So it's unusual, but what does that mean for you, the average astrobiologist? It means you can go to a place like Europa, which is one of these ice-covered moons of Jupiter, or Enceladus, which is an ice-covered moon of Saturn, and you can have an icy crust that's enclosing liquid water. Because if it were the other way around, these moons would be frozen solid, and you wouldn't be able to talk about the potential for life. So anyway, there are all sorts of wonderful things about water. I'm not quite as convinced about water as I am about carbon, but nonetheless, let's just sort of assume that's the basic building blocks. And from there, you want to have these basic polymers, these chains that go to make up much of the cell. So you've got DNA and RNA and lipids and proteins. There's a way to get all of these components abiotically, so without biology. Lipids seem to be relatively easy. We found lipids in meteorites. Dick mentioned our colleague Dave Deemer at Santa Cruz. He's done a lot of work extracting lipids from meteorites and then making little vesicles. So these are coming in from space. Not very difficult, apparently, to make these fats abiotically. Those of us who are constantly battling weight know that it's not even very difficult to make them biotically. Proteins, amino acids were made in the Urey-Miller sort of experiments, and although the exact starting materials that Stanley Miller used, as Dick said, are no longer considered completely relevant. There are other experiments that are done like that with more relevant conditions, throwing in metals and so on, that are also showing amino acids. You find them in meteorites, you find them in space, and so on. And there's been non-enzymatic polymerization shown with heating and drying and so on. So we know that you can get proteins without life. RNA, it turns out, is a much trickier one. It turns out that it's extremely difficult to make abiotically. You don't normally see RNA. You don't even really normally find bases or these building blocks of RNA in meteorites, Urey-Miller sort of experiments, and so on. More recently, some of these have been made in the lab under fairly realistic conditions in terms of the early Earth, but it's not trivial. And then Dave Deemer again is showing, you're starting to wonder why you didn't just get Dave Deemer tonight, probably. He's shown that you can polymerize these RNA monomers into a chain, so it's sort of like taking beads and making them into a chain by wetting and drying them with lipids there. So it's not impossible to get RNA abiotically, it's just very difficult. DNA, it turns out, is even worse. So think back to this idea of evolutionist tinker. You only have what's available. You're starting to get a feeling that some of these are going to be more likely as the building blocks for life than others. So let me give you one other sort of clue to this, where I'm going, and that is the central dogma. This is something that you may well have learned about in high school or college. And the idea is that you have DNA, deoxyribonucleic acid, so this is this double-stranded, sort of your coding for life, what makes, has the information to make you you. And that material keeps that information about you, but then it's transcribed into RNA. So it's sort of like if you have a rare book library, you can't take the books out. So the best you can do is make a Xerox of the page you're interested in. Maybe this is, these are the plans to remake the Taj Mahal or the White House or whatever. So you take the plans, you Xerox them, you take them out, and then you make them into something, and that something would be a protein. Then the proteins in our bodies can make everything else, for the sugars and lipids and so on. So the central dogma is this DNA to RNA to proteins, and the proteins help make a replica of the DNA.
The innermost workings
So let me
of
show
how
you
a
a
simple
video
code
that
is
explains that
turned
again.
into flesh and blood.
What's very cool about this is it gives
This
much
is what
more,
Francis Crick
I think,
called
gut
the
feeling
central
about how this
dogma
happens.
of modern biology. How DNA makes protein. It starts with a bundle of factors assembling at the start of a gene. It's these that trigger the first phase of the process, reading off the information that will be needed to make the protein. The gene is the length of DNA stretching away to the left.
What's kind of cool is all these proteins gathering, getting ready
Everything's
to
ready
actually
to
make
roll.
the RNA.
3,
Ready?
2, 1.
Watch this. And they're off. And this is basically in real time. It
The
happens
blue
very, very
molecule
quickly.
racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the two strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to make an exact copy of the A's, C's, G's, and T's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related nucleic acid known as U. You are watching this process, called transcription, in real time. It's happening right now in almost every cell in your body. When the RNA copy is complete, it snakes away from the nucleus and into the outer part of the cell. Then, in a dazzling display of choreography, all the components of another molecular machine lock together around the RNA to form a miniature factory called a ribosome. It translates the genetic information in the RNA into a string of amino acids that will become a protein.
So that's basically a central dogma. Is that incredibly cool? So, I mean, this is happening at an incredible rate in basically all the cells in your body all the time. So here's the central dogma, and we're left with this question of which came first, the DNA, the RNA, or the protein. And so this is sort of this chicken and egg problem that Dick was referring to. And the way an engineer might think about this is that RNA is the answer. Now, the reason for this is that RNA, as Dick said, does a whole lot of things. It can provide genetic material. It's got that information because, remember, even today, it's just making a copy of the DNA. But it also turns out that it can do some catalytic activities like a protein. So you think it's the answer to all life's problems. And again, Czech and Altman shared the Nobel Prize for this. I'm stumbling over this a little bit because I did actually have Sidney Altman for a class once, and I'll just leave it at that. Anyway, so that's the engineer's answer to this, that you can have RNA. This is out of one of Andy Ellington's papers. So he's pushing this RNA first idea that you start with this RNA replicase, and he's calling it the stock price. Then you get translation invented, and so all of a sudden the price of proteins, the value of proteins go way up. But it turns out that there are even more things that we've discovered that RNA can do besides making proteins and having these enzymatic activities. They have all sorts of regulatory roles. So they can make aptamers, they can bind small metabolites and help control genes, silencing RNA, microRNAs. This field has just exploded in the last 10 years. So you sort of feel overwhelmed with RNA, and you begin to think that not only was there an RNA world, we still live in an RNA world. So there are all sorts of things in favor of it, the fact that RNA can be a hereditary material. There are a lot of cofactors and vitamins and so on that your body uses that are basic ribonucleotides. You make DNA, deoxyribonucleotides from ribonucleotides. The DNA is the template for the RNA, but the individual monomers are made the reverse way. And then you've got these ribose switches and all sorts of incredible things I just mentioned, including the fact that there's sort of a molecular fossil in DNA. And these are palindromes. And so what a palindrome is when you can read something the same forward and backwards. And so this is the classic one, Madam, I'm Adam. And if you read it backwards, it's exactly the same. So this is a palindrome. Now, since RNA also likes to be double-stranded, but it's normally single-stranded, it's also looking for palindromes so it can form a more stable structure. So if that RNA were the ancestor for the DNA, you would end up with DNA that has a palindrome in it, which really doesn't make a lot of sense any other way. So these are all just sort of little tidbits and clues that the RNA world is the way to go. And then you ultimately switch and you store your genetic material in DNA. It's more stable and so on. That's fine. And so here it is in a slightly different graphical form. You start with the prebiotic world, and then you form RNAs and use that as catalytic and genetic material. Then you start to make proteins. You have the origin of ribosomes and so on, and ultimately you add DNA in. The problem is there are a whole lot of problems with this view. And these include the fact that the nucleotides are more complex than amino acids. There are problems with these ribozymes, including the making a ribozyme that makes a copy of itself that can then split and make more copies. So it's great that it holds the genetic material, but if it can't use that for anything, and then it can't make multiple copies, you're sort of at a dead end. Again, it sounds great if you're an engineer. In practice, maybe it's not so good. These RNA polymers are kind of fragile. And to me, the real kicker, even though there are a couple more things on this list, is that there are far more amino acids produced abiotically than nucleotides, and so on and so forth, including the fact that you don't find nucleotides in the interstellar medium. So I have colleagues here at NASA-Ames who take telescopes and look up into the sky. So they're chemists that are looking for the chemistry in the interstellar region, so they don't need test tubes in the lab and so on. And the idea of doing chemistry, at that kind of distance is just incredible. And they see amino acids. They don't see nucleotides. So some of these amino acids overlap with the ones that we have in our bodies. And so you're sort of left with this idea that if life arose on a planet, say, around Alpha Centauri, those poor students are probably using the same textbook that I suffered through, except, it's an Alpha Centaurian or whatever. So there seems to be this uniformity in the basic language of life. So here are all the, more and more problems. So let's try to look at this whole question of which came first now, instead of as an engineer, as a tinkerer. And you can see where I'm leading. What you really have to play with are not what's the best idea, but what's simply available. And what's available are the building blocks of proteins. And so here is this RNA world idea, and here is a protein world idea, where you have these membranes that are produced abiotically. We know that because you can extract them from meteorites and form little vesicles. You have these amino acids that are produced abiotically, and in fact, Just today, I got another confirmation from a chemistry colleague of mine at the University of Glasgow that they've used Urey-Miller sorts of experiments and gotten amino acid chains with 2, 3, and 4 amino acids. And so again, just that kind of reflux system that Dick showed you. So you can start to form these proteins completely abiotically, and then you bring in RNA. And what's actually kind of interesting is the way our bodies make RNA, ribonucleotides and deoxyribonucleotides, is from, you guessed it, amino acids. So even if you just look at what we do today, we start with amino acids. And then I think we all agree that DNA was probably last for a whole lot of reasons. So I'm going to finish by talking specifically about what we're doing in my lab on this protein world idea. And this is really work that is the brainchild of Kosuke Fujishima, who's been with me for a couple of years now from Japan. We're extremely fortunate to have him here. And so rather than an RNA world, we're saying we have an amino acid protein world. There is your basic stripped down amino acid. It's got an amino end and it's got an acid end. The R can stand for a whole lot of different things because there are about 20 amino acids that we commonly use in our bodies. There are another 2 that are relatively common. And then there are about another 100 that bacteria and so on use as odd things in antibiotics and so on. So when people talk about only 20 amino acids, that's not really true, even in life on Earth. There's a fairly broad repertoire, but they all have this basic structure. So R can be a whole bunch of things, but basically they all have these amino and acid ends. Now, I'm sorry that animation went a little faster than I was planning to show. This is from a paper that was published by some Canadian colleagues of ours at McMaster, Higgs and Pedratz, a few years ago. And what they did is they looked in the literature about making amino acids using a Urey-Miller sort of experiment, looking in meteorites, looking in the interstellar medium and so on, and then just put together the statistics. And what you find is that there's a pecking order. There are about 5 amino acids that you find over and over in fairly high proportions: glycine, alanine, aspartic acid, glutamic acid, and valine. So those are the ones I have in light gray up there. Over and over, these are the most common ones found. So you think, aha, you know, evolution is tinker, this is what we have available. And then you see about another 5 or 6 that are relatively common, and beyond that, you don't see them at all, because the rest of the ones that are used by life on Earth are then synthesized by life from these original ones. So what we're trying to do is take this fantastic 5, as we call it, and see what we can do with that. So if we're assuming that what's available in our workshop as a tinkerer are these 5 amino acids, what sort of capabilities can we build with this? And so at this point, Kosuke and various other students have been working with him, have started to put together short, relatively short, about 50 to 100 amino acid small proteins to see what sort of capabilities they can re-evolve, and then they're also moving to the 10. Now, the reason we think this strategy will work within our natural lifetime, which basically means before the next grant proposals due, is because there is a professor at Princeton who's doing a similar sort of thing with all 20 amino acids for completely different reasons. But he has been able to re-evolve capabilities. So he's evolved them, but it's a completely different origin than the capabilities that we have today. So certain enzymes and so on, for example. So we're doing a similar thing, but we're saying we only can use what's available, these amino acids that you would find in these prebiotic settings. So we need to look for things like binding to nucleotides. The metals are incredibly important, as Dick said. We realize that not just for the proteins that we're doing, but even in these more recent experiments synthesizing ribonucleotides, the whole key was having phosphorus in the whole time, whether you need it or not. Something like 60% of the proteins that life uses are metalloproteins. So we really are metallo-organic composites, if you want to think of ourselves that way. metals are extremely important. So using the metals, using those amino acids, let's see if we can bind the metals, let's see if we can bind nucleotides, let's see if we can make them, let's see if we can unzip then RNA, if we can get that far, ligate it, acylate it, get fatty acid binding, so in other words, membrane binding, phospholipid synthesis, peptide binding, see if you can make bigger and bigger proteins. So there are all sorts of things that we're now poised to do. So again, this doesn't prove that life originated this way here, or that if it ever arose on Europa or Enceladus or all these wonderful places that Kepler's finding, that it occurred this way, but it gives you a plausible route, not just on Earth, but if there is really this pecking order of amino acids out there, it should give you some kind of universal concept for how easy or difficult it is to form life. And so I think at this point I'll leave it there, and thank you very much. And I guess at this point we get some questions. So now you can tell me everything I said wrong.
Not at all.
Thank you for your insights, Dr. Rothschild and Dr. Zare. Now is the time for some questions. I've been asked to get the ball rolling here, but if you do have a question, remember, just please raise your hand, and our microphone-wielding volunteers will approach you. My first question here concerns recent discoveries of the Kepler space probe. And would either of you like to comment on what Kepler has discovered in short form, and the significance of that for our question?
Go ahead.
Well, we're sitting at NASA-Ames, and for those of you who don't know, Kepler is a satellite mission that's just recently put out its lights. That is one that was actually proposed and run by NASA-Ames, which is very unusual because usually we come up with the great ideas and then the Jet Propulsion Lab builds them and is in the news and stuff. So we're very, very proud of Kepler. And all it was, I hate to say was, it's like talking about Dear Departed, but it really is, was a very simple idea, and I mean that in the nicest possible way. All it did was stare at 150,000 stars. The only time it blinked was when it turned around to send data back to the Earth. And what it was looking for is slight diminutions in the light. So the idea is it was looking to see if a planet was passing in front of the star, and particularly looking to see if it was periodic. And so, of course, that depended on the planets being in a correct plane and that you could actually pick this up and so on. And it turns out that was a magnificently successful approach. And so when this started a couple of years ago, we knew of about 200 or so extrasolar planets. We now have literally thousands of extrasolar planet candidates. And if I'd given another talk, I have lots of figures, and you can see the whole planetary systems. What they're specifically looking for is Earth-like planets. So if it's planet's too big, you end up with a gas giant, and that's not going to be a good place for life to form, although it could conceivably form on a moon around it. If it's too small, you lose your atmosphere and so on. That's sort of the problems with places like Mercury and Mars to a large extent. And so they're looking for things within a certain size range that would overlap with the Earth. And again, they've literally found hundreds and hundreds of candidates. And this is only looking at 150,000 stars, which is a tiny sliver of the Milky Way. So what it suggests is that there are many, many places, not just even in our galaxy, but probably all over the universe, that have at least the basic components that you'd need for life. Now, whether they have volcanoes and the right phosphoric salts and sulfur and so on, Dick will have to tell you. But at least it's a start. At least we found a lot of places that at first blush appear to be habitable.
All
I'd
right.
like to,
Yes, please, Dick.
if I might comment a bit about those too. There's a sort of wonderful poem, if I can remember, that goes something like, big fleas have little fleas on their backs to bite them, and little fleas have littler fleas, and so on ad infinitum, right? And just as we have moons around planets, we're finding we have planets around stars. And we also find that there are stars that also revolve around other stars, binaries and such. So, and we now recognize this, because of Kepler, as being common rather than extraordinary. Yep. Before then, it was thought to be extraordinary. That's the change.
I have to say, I'm the oldest of 5 kids. My youngest sister worked for Nickelodeon Online and a few other places, and I think that there was a certain feeling, not her, of course, but among her colleagues in New York, that these things that they could dream up and the fiction that you see on TV was far more fantastic than real life. But the answer is that real life is far more fantastic than anything you could dream up in, one of these studios. It's really amazing.
Do we have a question? Let's see.
Yeah, I have.
Maybe up here?
So,
Oh, sorry.
the tube worms offer an intriguing possibility. My understanding is their basic biology is based on hydrogen sulfide, with sulfur being the oxidizing agent rather than oxygen. Doesn't that open the possibility to truly different kinds of extraterrestrial life on planets that we are now thinking, should be devoid of life, when in fact they might be rich with a completely different kind of life?
Can I disentangle that for a moment? And I say that because my husband's about 5 seats down from you, jumping out of his skin. That in fairness, I'm looking at you, bacteria have far more metabolic diversity than us eukaryotes, multicellular. So the tube worms are a nice example, but it's their bacteria, and that's just one out of lots and lots of examples of bizarre metabolisms that are already available on the Earth. And yes, it does open up quite a bit more places where you could potentially find life. Is that okay?
Dr. Rothschild, I was wondering if you could speak a little bit about prions and how they may or may not fit into your idea of an early protein world.
Funny you should mention that, because I've mentioned it a couple of times to colleagues, and they just sort of scratch their head and say, I never thought of that. But the big problem that people bring up with the protein-first world is the question of heritability. And that's why I was thinking of prions, these little protein bits that basically force other proteins into the same conformation. And so I don't know, that's how far I've gotten in my thinking, which is just scratching the surface, but I agree that there could be something interesting there. Even if prions are much later, it gives sort of a mechanism. Do you want to say anything about prions?
No, but I do like the idea very much that we start by thinking about amino acids first in that part. But amino acids, you have to be careful when you talk about a world. Amino acids or peptides or proteins, they're not alive. At least I don't think of them the way we've defined life, right? And again, we're trying to find that recipe, which seems that how do we get from? I'm back to this idea again that I don't want to just concentrate on either the car or the road. I need them both to make a transportation system. And somehow I need to bring together some things from this. And it's driving me again to think I need reactors, microreactors, in which I have amino acids, but I have to have metals. I have to do more things to it to make it happen. And the point is, we can now carry out these experiments. I think they're really worth trying.
Where's our microphone, please?
I would like to ask 2 questions, if I may. First one.
It'll cost you twice as much.
Oh, no. You'll have to keep the answer in the same time, I know. But I believe you said, if I heard right, that we're seeing with our telescopes amino acids in space, and I would love to know how we can actually see something as tiny as an amino acid in space.
I can explain that too.
Go.
Let me just ask my second question, because then I can give my mic away.
Yeah.
I would love it. I know you're both scientists and will not want to answer this, but I would love it if you could tell us, based on everything you know, and I know it's an open question, if you had to put your money down, are we alone?
I think my answer to that is, my understanding is that about 10 years ago, Lloyd's of London stopped taking bets on whether there was life on Mars. And if they've stopped taking bets, I'm not taking bets either. The thing is that if what we think about the origin of life, that there's a certain chemical sequence, as you can see, I'm trying to focus on things that would be universal, not every single body, but that are fairly common in terms of the most thermodynamic stability, what you actually find, and so on. If what I think is true for the origin of life, it would be incredible if we were really alone. But if we were, it means we're missing something, and that would be an incredible conclusion. Now, within our lifetime, we may find that we're not alone, but we won't know for sure if we really are.
Let me try.
And the other question is the spectroscopy one. That goes to
the other one is a spectroscopy one, and it's based on the fact that certain molecules absorb or emit light that's characteristic of that molecule. That's how. And it turns out in the microwave region, it's like a fingerprint. And people go in the lab and they measure these molecules, and it really belongs to that molecule. And they look, and just as they look and they see the elements in the sun that they find on Earth, they found now in outer space molecules the same as they find on Earth. That's how they know. It's radio astronomy, in other words.
Oops. If the light needs to glow green on the button near the button.
Who wrote that one?
I'm sorry, I don't remember what the it had rather religious title, Genesis. Karen Smith?
Yeah, that's what I suspected. Okay. Do you want me to go ahead and answer that? Okay. Yeah, Graham Karen Smith is a...
Repeat the question.
Oh, okay.
Let's repeat the question.
It had to do with the potential of having a replicating crystal. And I actually know Graham Karen Smith pretty well. He's a retired chemistry professor from the University of Glasgow. I do have friends in other places besides the University of Glasgow. It just so happens these two guys are there. Whereas my kids are at the University of Edinburgh. But nonetheless, Graham has pushed for many years this idea that you could not just use things like clays or crystals as a way to concentrate the building blocks of life, sort of more like what Dick was suggesting with the granite, but also that clays, for example, template so that the next molecule in line is lined up along a certain structure. Mm-hmm. And therefore, as they get bigger, it's like genetic information, and if you then fragment this crystal, it keeps growing. And so this would be an analogous to, well, more than analogous, he thinks homologous to an early life. And so I think in Dick's terms, that would be like not thinking of the transportation system, did the cars come first to the highway, but an evolution from footpaths to highways, that you start with a replicating crystal and ultimately organic molecules are substituted for the crystal parts.
I want to comment that there is some, what shall I call it, element of truth to this, that it's not just simply crystal. It's this idea. People have talked about the possibility that a wind would blow through a junkyard and assemble a Boeing 747 airplane. And they said that's so improbable, that's why life, I mean, this is all nonsense. You're wasting your time, I'm wasting my time talking about this because it can't be that way. And what's forgotten in this argument is it's not just probability of randomly putting things together. There's a concept of what's called self-assembly. Things order themselves as they come together and grow. And this is really the element of truth, I think, in what he's saying. Yes. Whether it's mud or crystals or whatever, there has to be some organization that's going on. And otherwise, indeed, it is like wind going through a junkyard. You don't believe it.
Early on, why was there catalysts necessary? Why were catalysts necessary? We had 1 billion years, and if there's just concentration and time, why would you need early catalysts?
my own picture in terms of my understanding of chemistry is that unless I get things to speed up reactions that will go, they just take too long, even billions of years, to do all the processes that are necessary. So I'm looking always to find some catalytic step. I might mention that right now in industry, the chemical industry is all based on catalysis, pretty much.
Yeah, and in fact, it wasn't billions of years. We have good evidence for life well within 1 billion years, and in fact, probable, I think, even within 100, 200 million years. And it may have been very quick. We don't know how long it took. I heard just a few weeks ago of a very sort of interesting paper, which I haven't read, but it's gotten me thinking about that very question, that many of these reactions, like we were talking about these polymerization reactions, need to occur at fairly high temperatures, but there are other things that need to occur at low temperatures. And it was that drove the evolution of these biological catalysts, because you had to be able to bring the temperature down, but if you bring the temperature down, you slow down the reaction too much. And so, I'm not for or against that, but I thought that was an interesting perspective.
Right.
We have time for 2 more questions, then I've been asked to give the final question. So, yes.
With respect to finding life elsewhere in the universe, it's my understanding that you can look at the atmosphere of planets, things of that nature, and oxygen is one of the markers that you look for. So my question is, with Kepler, what's the technical capability today of interrogating the atmosphere of other planets, and what can you really determine from that?
Well, there are 2 parts. One is it's not just oxygen. What you're looking for is a disequilibrium. So, for example, our atmosphere is 21% oxygen, and most of the rest is nitrogen, and that's a really unstable situation. So, for example, Enceladus, this little moon of Saturn, is spitting out oxygen, but it's not because of life, it's because water is coming out that is then broken into hydrogen and oxygen by the radiation coming from Saturn. So you can have the oxygen, it's the unstable combination. Kepler is not really looking for the atmospheric compositions, but that is the big question. I just had a visiting grad student from the Max Planck Institute who specifically was asking that question. So Kepler's found all these candidates, there are other telescopes doing the same thing. What are you going to do about it? You're not going to go there next week. And it's very frustrating. So the next thing that you have really is the spectroscopy, is to try to get an idea of what's in the atmosphere. And it's even worse than that, because you may only have 1 to 4 or 5 pixels for the entire planet. So can you imagine if you were, X light years away and all the information you had about planet Earth was 1 to 4 pixels. So you try to extract as much information as possible.
It's very, very tough. And first of all, if you look back at early Earth, you find out it didn't have oxygen. Actually, oxygen came with cyanobacteria. It came, right?
Yes.
Okay.
Oh, my friends.
My friend, my friend too. Okay. And so you would say, oh, our Earth is dead, but it wasn't dead. It was alive, and we think. And the other thing is, suppose you look at a flame now. If I had a lighter, I'd like a flame. And what's its color? It's yellow, right? And suppose you go away and you look and you analyze it. You'd conclude it's made of sodium. But the flame has very little sodium in it. But that's the thing that's dominant to your eye, to what you see. And we must realize some of the limitations of spectroscopy here.
That's neuroscience.
As well. and so it's going to be tough. But this equilibrium, being out of equilibrium, is very important. But we know that radiation can cause disequilibrium, as you pointed out. So it's tough.
So I think it's more the frustration that you feel like you've got to do something next without going there, and that's what seems to be what we are able to do. It's not that that's ideal, but that's what we can do next.
But the technology is such that with a limited amount of information can actually draw some conclusions about that.
Yeah, because as Nick said, that different elements have, and different molecules have different signatures. So when you
really
get a
have
spectrum,
different colors.
Yeah, exactly.
Young man.
Why did it take billions of years for single-celled creatures to evolve into multi-celled creatures, but it took only a couple million years? Like, let's say cells started about 3 billion years, and the first jellyfish-like stuff came in 1 billion years. So it took 2 billion years to get out of the bacteria stage, but it took less than 500 million years to get into primitive life, like dinosaurs, rats, mice, people.
Yeah.
But it took billions of years to go from 1 cell to 3 cells.
You know what? I don't understand that either. And that's why I don't think the fossil record is right on that point. It makes no sense to me whatsoever that you would get the origin of life like that and then sit around for 2 billion years before you have a eukaryote. I completely agree with you. And I'm not the only one. So I think when you go back to the fossil record and try to understand when you see a little dot or a scratch in a rock, whether that was a bacterium or was it a eukaryote, or did it have more than 1 cell? We have very, very strict, we paleontologists have very, very strict criteria. So it's got to be huge, and it's got to look like this, and it's got to be that. So they're missing a lot. So I actually believe that that's exactly, that point, that the eukaryotes and probably a certain number of multicellular ones were very early. We just haven't picked them up in the fossil record yet.
It's easy if you find a chicken bone, but otherwise you're in trouble.
My last question is very much like yours. The SETI scientists, those who conduct the search for extraterrestrial intelligence and have done so for 40-some years, have come up empty so far. But they love to point out that it's a big cosmos they've just gotten in the game. Still, I'm pretty sure they're sad that we've gotten no signs of technologically advanced life that we can detect anyway. And I'm wondering is it possible that, as that young man suggests, that after this remarkable finding of Kepler, that there may be billions of Earth-like planets in the galaxy, and that is the catch perhaps, if not the origin of life as we're discussing tonight, is the catch this evolution of complexity?
Oh, deep questions we don't know the answer to. Why is it that the dinosaurs disappeared, right? If they hadn't disappeared, when they were around, they seemed to be dominating life on Earth, right? They were the king. And then they disappeared. And we can talk about what happened, why we think they disappeared. But this thing about intelligent life, I'm not sure there is any intelligent life, including here.
Ouch.
I have the trouble, you see, that I just recently came back from China, and they're asking me, how is it possible that your government shut down for 17 days? They can't understand this.
Well, no,
and I'm having trouble explaining that.
I've hung around enough with the SETI people that you should understand that the way the SETI people define intelligence is anyone who can build a radio transmitter. So with that definition, how many people in this room would consider themselves intelligent? Because I'm out of the game. So they are looking for very specific sorts of signals that if we had a world full of Shakespeares, they would not pick up intelligent life on the Earth. Could be Shakespeare, Newton, and Darwin as far as the eye can see, but you wouldn't pick that up with the methods they're using.
I hope you've enjoyed this evening's program. Yikes. Brought to you.
Thank you.
Brought to you by.
Great.
Brought to you by the Commonwealth Club and by Wonderfest. And I hope you'll join me, as you just started to then, in thanking our speakers and thanking NASA for letting us be here and thanking your fellow audience members. Great questions. Very good. Thanks very much.